Hello everyone, my name is Christine Boy and I am a staff scientist at the United States SLAC National Accelerator Laboratory in California, funded by the Department of Energy. And I am very honored to be presenting our work on how some things that are often overlooked, meaning small-scale processes and variability, that still may influence large-scale environmental processes and outcomes. And as you all know, it takes a lot of exceptionally talented scientists and supporting staff to do environmental research. And so here are some of the fabulous contributors of what I will present today. There are many more that worked hard to contribute to all of this work, but it would take the entire presentation to acknowledge them all, so I will refrain from that. But I do want to especially highlight my dear colleague Vincent Noel, who has been my partner in crime, so to speak, from the start of these investigations, and he has been and continues to be a really foundational driver of the science that I am presenting here. So let me just set the scene briefly. Our work is focused on floodplains. So that would be alluvial aquifers and also how they impact the groundwater and river water quality below them and next to them, which is exemplified by this work by Jeannot et al., which is a reconstruction of anoxic sulfur accumulating zones that strongly impact uranium mobility in a contaminated, uranium-contaminated floodplain. And so what I really want to highlight here is that floodplains are highly heterogeneous environments due to the variation in sediment deposition from the rivers and the changing hydrologic conditions over time. And so sediment texture and organic matter content varies in depth, but also strongly controls the hydrogeochemical behavior post-deposition. And so the findings by Jeannot et al., among other papers, led us on a path to further understand the dynamics and controlling factors of the impact of, in this case, anoxic anomalies in predominantly oxic floodplain subsurface environments. So through multiple rounds of column experiments, we found that the insert of a small fine-grained, organic rich lenses in oxic coarse-grained matrix, these lenses did not only just remain anoxic, thereby resisting the continuous influx of oxic water, but they actually spread the anaerobic reactions to the coarse-grained matrix downstream, and that included sulfate, arsenic, and iron reduction. And so that was kind of surprising, as we had hypothesized that the lenses would actually be oxidized, but instead they spread their anoxic conditions. And furthermore, we found that the solid phase changes in the coarse matrix could not be explained by the dissolved exports from the anoxic lenses. And this was further demonstrated by reactive transport modeling that considered both the aqueous and the solid phase data, and that's important. The model suggested that particulate organic matter transport that was not observed in the aqueous samples must have occurred to support the observed downstream changes in aqueous and solid phase chemistry. And we also have some preliminary 16S sequencing data that indicates that this transport may have included live anaerobic microbes, or they were dormant in the coarse-grained matrix and then became active because of exports from the fine-grained zone. And then in a separate modeling study, which was at the scale of tens of meters rather than tens of centimeters, we used DGSA and found that the down gradient spread of reducing conditions was controlled by sulfate reduction and dissolved organic carbon release from these lenses, and also that the total volume of fine-grained lenses was important, but their individual shape and size did not matter, which further indicates that these are diffusion-controlled exchanges, which was suggested by our column studies. And then sensitivity analyses from the same study suggested that the fluid residence time, which is nominally controlled by sediment permeability, was the most controlling factor for export concentrations. And so therefore there is a need for more precise permeability measurements at the field scale in order to understand how these fine-scaled heterogeneities influence larger scale hydrology and exports. So all of these studies led us to develop a three-pronged research program to better understand and thereby predict floodplain biogeochemistry and environmentally relevant exports. And so this involves quantifying and characterizing small-scale subsurface heterogeneities in sediment composition. And as just mentioned, more precise information of permeability and fluid residence time, and also to quantify colloidal transport, which is trickier because it also requires determining colloid composition and reactivity, and yeah, there is a lot of work to be done there, which I will get to towards the end of this presentation. So for the small-scale heterogeneities, we have developed and utilized a framework that is based on two-dimensional X-ray fluorescence mapping to identify and quantify differences in elemental concentrations and speciation in, for example, soil cores or greenhouse rhizoboxes. The results clearly indicate that anoxic microsites are present in all soils that we have examined to date, whether that's like on a floodplain or a toe slope or an upland soil. And I also want to highlight that this framework can also be used for identifying other types of heterogeneities, whether they are redox or otherwise, in a wide range of samples, such as plants, organs, paintings, etc. So that was a very, very brief overview of our analytical platform for X-ray identification and quantification of small-scale heterogeneities, which we call LOMES. I will spend a bit more time on the second prong of our research, which is basically about hydrology. So I will take you to our most heavily examined floodplain, which is located by the Slate River in Colorado, United States. It's an alpine environment that is one of the headstreams for the Colorado River, which supports a large part of U.S. communities, farms, and water power facilities. And so although our floodplain study site is pretty small, it still contributes to large-scale impacts, and we're using this because it's a good place to understand the mechanisms of subsurface processes, which I will get to in a moment. So it's pretty small, and primarily it exhibits a sandwiched oxic-anoxic soil profile, as you can see on the bottom right, above a highly conductive gravel bed. And this gravel bed primarily receives the water from the river and not from groundwater, which explains why it sort of propagates oxic conditions into the fine-grained soil. And just to be clear, this is the floodplain site that we're looking at. So the reason that we have so heavily focused on the Slate River floodplain site is because we were so lucky as to have instrumented and started sampling the site in 2017 and 2018. And then in 2020, some beavers decided to build a dam in the fall, and that dramatically changed the hydrology and thereby the biogeochemistry of the floodplain. And then a couple of years later, in 2022, the river migrated due to some sediment deposition, and so the beaver dam actually dried up in July of 2022. So basically we had a field scale experiment sort of just given to us. So we've spent a lot of time trying to understand the hydrological influences and then how that impacted the biogeochemistry. And so one of the most important impacts of the beaver dam was the changes in vertical water flux, actually, which is shown on the bottom right for our 3 primary sampling locations, OBJ1, OBJ2, and OBJ4, and they're all, OBJ1, OBJ4, and OBJ2 with varying distances from the beaver dam. And so these vertical water fluxes appear to also be highly sensitive to the anisotropy or heterogeneity of both the fines and the gravel bed. And in the fines, there are specific preferential flow paths created by coarse-grained pathways or lenses, or whatever you want to call it, within the fines. And so the dynamic hydrology at this field site, due to the river discharge and the beaver dam and the heterogeneity of subsurface sediments, sediment composition and permeability, is obviously highly complex. We see strong dependencies on river discharge and the ponding and flooding extent, and flux directions or flow directions are varying over a season. However, when put into context of other floodplains studied by our group, and scaling sort of the export to soil, from soil to groundwater, or gravel bed water, it appears that the ratio of lateral flow in the gravel bed to the vertical flow, so called that the infiltration rate, if you wish, that scales along a logistic or S-shaped curve, even between seasons and across different size floodplains. And so that's encouraging in terms of finding more general patterns. And we've also found that the exchanges, the vertical fluxes versus the lateral fluxes, has strong implications for the biogeochemical processes and the element transport, which is indicated here by these statistical model simulations of specific conductance and oxygen concentrations relative to horizontal flow in the gravel bed over time. And then moreover, the concentration of iron 2 containing colloids are correlated with both the vertical pore water velocity and the dissolved organic matter concentrations. And this further accentuates the need to understand colloidal impacts in these systems. And that brings us to the final prong of our approach to understanding the subsurface biogeochemistry of floodplains and how it relates to groundwater and surface water quality, which is the colloidal component. And so, although colloids have been observed, considered important, and studied for multiple decades, there is still a lack of consensus understanding of their actual importance for biogeochemical processes and elemental transport of relevance for groundwater and surface water quality. And so to address this, we have developed workflows, experiments, and modified analytical procedures dedicated to identify, quantify, and characterize colloids in natural floodplains and river waters. And we have specifically focused on redox sensitivity and reactivity because that is lacking in current understanding of colloidal literature. And so the aim is to provide numerical parameters to include colloids in a chemically reactive way, as well as the more common physical filtration type of interaction with the solid matrix and water chemistry, flow rate, etc. So we have demonstrated that in our Slate River floodplain, again, iron colloids appear to be protected from redox fluctuations, so reduction oxidation processes, by organic matter and silicon coatings. And the organic matter composition of the colloids of various sizes and physical structure also vary distinctly between seasons and depth profiles. And so that has led us to develop some lab experiments to more intricately look at the reactivity of colloids exposed to varying redox conditions. And so from those very recent laboratory simulations, we are looking at the role of organic matter for preserving iron colloids, and it appears to be substantial. So next, we are hoping to develop some column experiments, etc., to look at the interaction of colloids with the solid matrix and the hydrological, physical, chemical, and biological dynamics to better understand the transport of colloids. So all of this is still very much a work in progress, so stay tuned for more findings. So for now, I will just finish up with a brief summary of what I presented here today, including the importance of colloids and particulates for driving downstream biogeochemistry and water quality, and the multitude of factors that control small-scale impacts on large-scale modeling and remote sensing observations. And with that, I want to thank you for your attention. I want to thank all of my collaborators again, and of course our main funder, which is the Department of Energy Environmental System Science Division. Happy to take questions. Thank you.